Welcome to the audio slides to our recent paper on development of kit formulation for radiolabeling of the minigastrin analogue CP04 with indium-111, aimed to be used in the phase 1 multi-center clinical trial within the AeroNet project grantee MTC. In our paper, we describe the chemical and pharmaceutical development and characterization of a freeze-dried kit formulation for radiolabeling of the radiopharmaceutical precursor CP04 with indium-111 as part of the investigational medicinal product dossier for the first-in-human clinical trial of this radiopharmaceutical. Medullary thyroid carcinoma is a neuroendocrine tumor of the C cells of the thyroid and represents one of the most challenging cancers for both physicians and patients. Patients with medullary thyroid carcinoma, or MTC, can be cured only by complete resection of the thyroid tumor and any local and regional metastases. Since often the disease has already metastasized at the time of diagnosis, many patients with progressive or metastatic disease cannot undergo surgery but are candidates for systemic therapy, which unfortunately has not demonstrated any improvement in survival of MTC patients. Nuclear medicine could play a major role in the management of MTC patients by targeting the CCK2 gastrin receptors on the surface of MTC tumor cells. Radiolabeled gastrin analogues with high affinity to these G protein-coupled receptors can internalize, prolonging the residence time of a radionuclide at the target site, leading to diagnosis and therapy depending on the radionuclide used. From a series of gastrin analogues, the indium-111 labeled peptide CP04 displayed excellent characteristics such as high stability, receptor affinity, specific and persistent tumor uptake, and low kidney retention in animal models. Therefore, it was selected for further clinical evaluation within the AeroNet project grantee MTC. Before starting a clinical trial, the radiopeptide had to be formulated into a radiopharmaceutical kit. After ensuring GMP-compliant synthesis of CP04 and testing of its impurities, a set of pre-formulation studies was conducted. The kit formulation was optimized by adjustment of buffer, additives, and radiolabeling conditions. Quality control and stability assays of both the kits and the resulting radiolabeled compound was performed. For the first-in-human study, 2 dosage forms are required: a 10 microgram CP04 formulation for initial safety studies and a higher dose of 50 microgram CP04, both to be radiolabeled with 200 to 250 megabecquerels indium-111 for patient application. Selecting an ascorbic acid buffer of pH 4.5 allowed freeze drying of the kit formulation with satisfactory pellet formation. Addition of methionine and gentisic acid as well as careful selection of radiolabeling temperature was required to avoid extensive oxidation of the MET11 residue in the CP04 sequence. Trace metal contamination, in particular zinc, was found to be a major challenge during the pharmaceutical filling process, in particular for the 10 microgram formulation. The final formulations contained 10 or 50 micrograms CP04, 25 milligrams ascorbic acid, 0.5 milligrams gentisic acid, and 5 milligram L-methionine. The radiolabeling performed by incubation of 200 to 250 megabecquerels indium-111 chloride at 90 degrees Celsius for 15 minutes resulted in reproducible radiochemical purity of greater 94%. Kit stability was proven for greater 6 months at 5 degrees Celsius and 25 degrees Celsius. The radiolabeled product was stable of greater 4 hours at 25 degrees. A promising radiopeptide targeting the CCK2 receptor was successfully developed into GMP-grade kit formulation to prepare the radiopharmaceutical indium-111 CP04. By additional substantial preclinical biodistribution and toxicological study, the investigational medicinal product dossier has been completed. The described formulation will be used in the first multi-center clinical trial in MTC patients with the aim to contribute in the improvement of diagnosis and therapy of metastatic MTC. Thank you for listening.